Hello, Life Science. This video is about the central dogma, which is a really important concept in cellular biology, and it's really a continuation of what we've been learning so far about DNA and chromosomes and genes and all of that stuff. This video is actually going to be broken into two parts. This first part is going to focus on replication, and then the second part will focus on transcription and translation. So, review of what we know so far. So far, we know that all living things are, all living things are made of cells. And inside our cells, because we are living, so we are made of cells, inside our cells are nucleus. So every cell has a nucleus, and inside every nucleus is our DNA. And our DNA is put together into a special shape called a double helix. And that shape is made because our DNA is composed of these bases. We have four bases that make up our DNA. We have adenine, which is represented by an A, thymine, which is represented by a T, guanine, which is represented by a G, and cysteine, which is represented by a C. And so in that DNA structure, the rungs of the ladder, if we're going to think of the DNA structure like a ladder, the rungs are made up of two of the bases, and they pair in a very specific way. So A always bonds with T, and C always bonds with G. So these rungs of the ladder will either be an A and a T, or a C and a G. And then they're twisted into the double helix, and then the double helix is twisted around these proteins called histones, and then those histones are, with the DNA wrapped around them, are coiled into chromosomes. And that's really just to save space so that they can fit into the nucleus of our cells, because there's a lot of DNA that has to go into each nucleus of each cell. Okay, so with that background review, let's move on to replication. So replication is a super important process where DNA makes a copy of itself. Why would DNA need to copy itself? Well, in multicellular organisms like our cells, we need to keep producing cells, like skin cells shed. And so we're going to keep producing skin cells to replace the shedded skin cells. And how that's done is a single cell will basically go through a process of division where it divides from one cell into two cells. And that's a process we're going to learn a lot about later, cell division. But why we're talking about it right now is because when that one cell divides into two cells, that one cell has to make an exact copy of all of its DNA, so that when it divides, there's one copy of DNA for the original cell and one copy of DNA for the new cell. And that process is called replication. So these pictures kind of go through the process of replication. So let's kind of talk through that a bit. If we have our DNA here, it's not twisted in this picture just because it makes it easier to see. But we have that DNA shape, that ladder-like shape. And you can see the bases are paired correctly, A with T, C with G. And what happens in replication, the first thing that happens is the DNA unzips. And it really looks like a zipper unzipping, but basically what that means is the pairs of bases split or break. So the A becomes separated from the T, the C becomes separated from the G, and on and on and on. So that each side of this ladder structure is separate. And then new bases attach to each side of the original DNA, still with the same base pairing rules. So A with T, A with T, C with G, C with G, and on and on and on and on, until you have an exact copy. And you can see here CG, CG, AT, AT, GC, GC. You have an exact copy of your original DNA. So instead of one DNA, you have two DNA molecules. And that's replication. So let's work on this a little bit more. If you have this DNA sequence here. First thing that would happen in replication is that it would split. So you'd have one side of the original strand separate from the other side of the original strand. And what I would like you to be able to do is to demonstrate replication by pretty much just showing those base pairing rules. So after the strands have separated, I'd like you to be able to show me what bases pair with those original strands. What I mean by that is if you have an A T is going to bond with that A. If you have a T, an A is going to bond with that T. If you have a T, an A is going to bond with a T. If you have a C, a G is going to bond with it. And you have a G, a C is going to bond with it. So really, I'd just like you to be able to show me those base pairing rules. So you can pause for a moment and go through this and show me what bases would pair with this original strand and which bases would pair with this original strand. Oops. Whoa. So, if you've unpaused now, check your work. With the top original strand, you would get this new strand with this sequence. And with the bottom of the original strand, you'd get this as your new sequence of DNA. So as you can see, these are two copies of the exact same sequences of DNA that are representative of the original DNA. Okay, let's keep working on this. So on your own, and this is, I'm asking you to do this because I'm going to ask you to do this on quizzes and tests. If you were given just a single part of the DNA, so instead of both sides of the ladder, if you were just given one side of the ladder, it would look like this. And to show replication, I would really just like you to show what pairs with these bases. Like we did before, whoops, the A would bond with the T would bond with the A, and then we'll skip this one because it's the same, I guess we won't skip it, A, and then a G and a C, blah, 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 blah. So take a couple minutes and pause and pair what would go with each of these single strands or half of the DNA molecule. So what would go with this half, what would go with this one, what would go with this one. Okay. At this point, there's lied. One more thing to learn. There's another vocabulary word that goes along with replication called semi-conservative. And basically what semi-conservative means, having some trouble here, is that you have your original DNA. And in the process of replication, that original DNA is unzipped or separated into two pieces. And new bases pair with each of those sides, each of those separated parts of the DNA. So that when you have your two identical DNA molecules, so you've gone from one to two identical ones, each of the identical DNA molecules has half of the original DNA and half new DNA. So that semi-conservative describes in the process of replication how half, so half of the DNA is conserved, so kept from the original, and then half of it's new. So you really need to know what semi-conservative means. It will be asked on quizzes and tests. At this point, you really should know where DNA is located in our cells. Why does DNA need to be replicated? What are the base pairing rules? So which bases pair with which bases? The A's, T's, C's, and G's. And then what does semi-conservative mean? If you were a little bit confused on any of those points, you should go back and watch this video again. At this point, this will be the end of the first video, and